So just quick introduction, why we've been interested in anticoagulants, particularly potent ones, is that all heart surgeries, cardiopulmonary bypass, angioplasty, et cetera, requires rapid onset anticoagulants, and there are millions of patients worldwide, as you know, require these type of procedures each year. What leads to them is you can have a clot, a blood clot in the heart or the brain in the case of a stroke or some other place. And moreover, when you do these procedures, it also induces clotting by activating this cascade over there to actually cause and trigger more blood clotting. So to do these procedures, actually, you need potent inhibitors of this process to let you even do surgery on individuals or do interventions to try to go and correct the challenge. Now, it's probably most of you know the gold standard for essentially a century has been unfractionated heparin. So here on the left, again, discovered over 100 years ago, it's been used for heart surgery. It's actually allowed for the development of heart surgery, so we wouldn't have it without it. The way it works, it's interesting. It actually potentiates another factor, antithrombin, to inhibit thrombin, as shown here, or factor 10A. So it's an indirect inhibitor, so it's very potent. It inhibits multiple steps. Fortunately, it's reversible by an ionic interaction with another with a protein from actually salmon sperm called protamine. And it's extremely because it's so old, it's been made at huge scale, very inexpensive. Some people say hospitals are addicted to heparin. Unfortunately, with its use comes risk of bleeding. You can reverse it, but it's tough to dose. It's indirect inhibitor. You can actually consume your antithrombin and only have a relatively short half-life. Some people can have an allergic reaction to it. And we, again, become particularly interested in thinking about alternatives because it's very unsustainable how it's produced. And this came to light really because of an article that appeared in Lancet a couple of years ago that really pointed out that you may not know that heparin is made from the intestines of pigs, and it has a high carbon cost. And so it pointed out, and moreover, just also pointed out that it's made in pigs, and if we have a swine flu, like we are having avian flu, now could wipe out our anticoagulant source, because most of those pigs are all, as shown here, in Southeast Asia. And it takes, believe it or not, over 1 billion pigs a year to make the world supply heparin. And so when you start to think about those numbers, again, this article wasn't us, 1 kilogram of pig produces 6 kilograms of carbon. And so pork industry generates almost 700 million tons of carbon dioxide per year. And so this is, obviously a challenge for the planet and sustainability in the long haul. So we've gotten quite interested in thinking about alternatives that are synthetic and not manufactured from large animals, because we just have to do that for the future. I actually say heparin sort of like coal. It let us do amazing things. We're near Newcastle somewhere here. Let us develop heart surgery, but unfortunately, because of climate impacts, we have to think about alternatives. There have been new anticoagulants, I'm sure you know, made over the years. Some of them are direct anticoagulant drugs. This is the famous one here, Apixaban, also called Eliquis. It's about an $8 billion a year drug. So see advertisements for it all the time. So they've been successfully made anticoagulants. These are direct oral anticoagulant drugs, or heparin's injectable. The downside of them, well, the upside of them is they're direct. They inhibit the catalytic centers of their targets, but they're oral and they're not as potent as heparin. So they're used as preventatives, but they aren't potent enough to do cardiopulmonary bypass surgery. They're less immunogenic. Again, they're limited, all of these drugs, because they really are specific enough, and it's challenging to reverse them as well. So for this reason, we've, for a variety of reasons over the years, been interested in Aptamers as potentially anticoagulants. I just want to remind people a couple of properties of Aptamers on this slide, which I probably don't have to do for this audience, but just want to point out one or two of them. Again, they're high affinity, high specificity, as you guys know. But I want to point out their main mechanism of inhibition is shown here on the left-hand part of the slide. Is that they actually bind so-called exocytes on protein. And so what they do in this case is this thrombin, it blocks, a substrate, a macromolecular substrate from binding thrombin. It does not bind at the catalytic site or center in thrombin, so it doesn't block the catalytic function. These are good. They're not immunogenic. They're reversible antisense oligos, but they're limited in their potency whenever we've tested them. We've taken some of them to the clinic. and treated a few thousand patients, but what you see is none of them are as potent as heparin. So with this challenge in mind, we started to think, was there any way to make Aptamers more potent as anticoagulants? And at some level, we turned to Mother Nature and we thought, well, how has biology and evolution figured out how to block blood clotting? And it turns out there's a lot of blood-sucking organisms in the world. And if you look at all of them, they're very similar, do a similar trick to what leeches do, which make this peptide protein called pyridine. None of them, by the way, make a heparin-like molecule, or they don't make a small molecule drug. They all use this sort of strategy where they make a peptide that binds two parts of, in this case, thrombin, and it turns out that makes a very potent anticoagulant. I'm going to show you why here on the next couple slides. So it's a bivalent binding protein or peptide. Part of it binds the catalytic site, shown here in pink, and then it's linked through a short linker to an exosite binding peptide sequence. You can see the two of them have a moderate KD, so 300 nanomolar, and then 10 micromolar for the exosite binding domain. So if you look at them alone, but if you link them, now you have 10 picomolar binding. So suddenly, by linking these two things that are targeting both an exosite and an active site, and that's what we call exact inhibitors for exosite active site, you can get very high affinity. So we thought, well, maybe we could do that with an aptamer. So we knew here's an aptamer binding an exosite. Could we put a linker on it? We would just use the nucleic acid linker and then append it somehow to an active site binding inhibitor. And so to first ask this question, could you generate such a molecule and would it be potent? We actually took a well-known DNA binding aptamer which binds thrombin, HC22. We put a 7A linker on it and then conjugated it to a carboxylic acid on the bigatran, which is a small molecule direct thrombin inhibitor shown here in pink. And then so we wanted to ask again, so the aptamer is relatively high, 22 nanomolar, binds so-called exosite 2. But it's not a very good anticoagulant again, because it's not blocking the active site, and it's actually not blocking fibrinogen binding. In this particular case, the bigatran is a small molecule. It's an FDA-approved drug. Again, modest, relatively good affinity for a small molecule. And so then again, we put this poly A linker in between them. So how well does that work? Well, if you look at just the affinity, now you're down to an affinity of about 50 picomolar association constant when you link the two. If you compare that to just the aptamer alone, shown here in the middle, with just an amine at the end of the aptamer, or the bigatran with a mutant aptamer, you can see you're about at least 100-fold higher binding affinity by making the bivalent type of compound. So they work at blocking thrombin activity. The simplest way to do this is a fluorogenetic cleavage assay where you add a fluorogenic substrate to the thrombin of the enzyme, a protease that cuts that fluorogenic substrate, and you can say, do I inhibit that fluorogenic generation by adding increasing amounts of inhibitor? Here's what you see with the bigatran from the literature. It's a moderately good inhibitor, IC50 of 50 nanomolar. Again, used clinically. The aptamer, as I mentioned, by itself is not very good. However, if you mix them together, it's very similar to bigatran. However, if you add them together, you see about a 500-fold improvement of inhibition. Actually, probably maybe more than that. We're getting to a titration amount because we're going to such low concentrations here. So suddenly, at a much more potent inhibitor by linking the aptamer and the small molecule. And moreover, as I glossed over, but one thing that people that are interested in aptamers and anticoagulants, particularly us, is that you can reverse them by using a complementary oligo to the sequence of the aptamer. So we wanted to ask, could you reverse this effect by using a complementary oligo to the aptamer and part of the linker domain of the gene? You can see you can reverse that activity. And interestingly, you reverse it better than you see with the bigatran alone. We think that's because if you make the base pairing up to where the bigatran is, you probably have a steric clash and actually pop the small molecule out of the active site. So how well does it work in simple clinical clotting assay? So what you can see here is the exact inhibitor shown in red are very good. They're at least as potent as the aptamer, or more potent than the aptamer small molecule alone. You can see that in 2 of the 3 assays, they're actually comparable to high doses of heparin, 5 units per mil, which is what you use during cardiopulmonary bypass. They aren't as good as heparin in APPT assays, you can see. So they rivaled them, at least in those assays, and again, you can reverse this, just showing here in person, these assays using reversible antidote optical color. We were somewhat disappointed, I have to admit, that we didn't beat or were comparable to heparin, at least in that one assay. So we said, well, can we go to a second generation? After here, we decided to target exocyte 1, which is a different exocyte on thrombin X is where fibrinogen binds. It's actually where uritin binds, like I was saying earlier on. We made that aptamer. There's some advantages to it. It's shorter, which is an advantage for the reason we just heard. Again, it's slightly higher. Well, the complex is higher affinity than the earlier exact inhibitor, the first one made. And again, it has more stability even for a DNA product because it's a cheap quadruplex type of aptamer. Is it as good now? How does it compare to the first generation? You can see in all three of these assays now, like 0.5 to 1 micromolar. I can't remember, 0.5 micromolar concentration. You max out these APD, PPT, and ACT assays. So it's more than 1,000 seconds. Again, even in a very highly procoagulant assay called Aptamer, thrombin elastogram time, you're now maxing it out. And it turns out that's good or better than heparin now. So using sort of this approach, we think we have something that can rival heparin. It's actually more potent than the fallback anticoagulant used for heparin called bivalirudin, just shown in the black boxes. Moreover, you can reverse this as shown here. So the last two slides quickly. Can you use this in vivo? The way we normally initially look at anticoagulants is a mouse model we use in the lab where you expose the carotid artery of a mouse and you chemically damage it. And so the new thing that activates the cells there and makes a very thrombogenic surface. And what you do is put a blood flow probe downstream of that site where you damage it, and you say, can we keep the vessel patent when we damage it? And what you can see here, if you do this with heparin, the vessel remains patent. And what you can see is if you do this with just saline, all the vessels clot off. This is looking at blood flow shown here in red within 5 minutes. Bivalirudin helps some, again, which is one of the better anticoagulants out there, but it still doesn't keep this patent, whereas the second generation exact inhibitor actually can for the whole time as shown here. Should be toggling through here. So that was encouraging to us as an anti-thrombotic, so it can block thrombosis. In that type of setting, we wanted to ask in a even more clinically relevant setting. And one of them is where you need a very potent anticoagulant. It's in so-called ECMO for extracorporeal membrane oxygenation. That's when we're circulating blood in a critically ill patient for a variety of reasons, happening a lot in COVID, unfortunately. We do it when we do cardiopulmonary bypass. And so you're taking blood and you're passing it through this membrane to oxygenate it, and you're putting it through tubing, and all that's very procoagulant. And so we wanted to ask, could you maintain this circuit with the second generation inhibitor? And you can do it with heparin. And one of the ways you monitor that is just the blood pressure flow through the machine. You don't want it to go up. If it clots, it'll go up because you have that resistance. So you can see heparin over a 2-hour run, this type of ECMO circuit can keep the blood, human whole blood flowing normally the whole time. With bivalirudin shown in yellow, you can see over time it starts to form clots and the pressure goes up. And again, the second generation Aptamer does essentially as well as heparin in keeping the pressure normal. And then if you look at even the membranes, et cetera, for clots, you can see there's no evidence of them even by like scanning electron microscopy. So on the left is standard dose of heparin used in bypass surgery, 5 units per mil. You see there's no clots on these oxygenated membranes. Again, the second generation exact inhibitor is essentially clean, just like heparin. And then bivalirudin, you see lots of big clots, and that's why the pressure goes up. So it's comparable again, and more the heparin more effective than bivalirudin. So we think it's potentially an alternative to unfractionated heparin, which again we think we need. So to sort of summarize this set of studies, we would say that developing these exact inhibitors where we're linking aptamers binding exocytes to small molecule drugs that stick in active sites, you can become an extremely potent inhibitor. Again, Mother Nature figured this out long before we did. These fortunately don't require an endogenous factor like antithrombin, so you're not worried about depleting it. So dosing and everything should be more standard and easy theoretically, and again, it's reversible by again this antidote approach that using, taking advantage of electric based pyramid. And so ongoing work, we're doing lots of pharmacology and pharmacodynamics. Maybe not surprisingly, you see extended pharmacodynamics compared to PK because these things bind so tightly. We're looking at toxicity and immunogenicity. We've never seen those things with regard to the Aptamer alone for anticoagulant, but again, we're sticking with small molecule in there now. Who knows what's going to happen? We don't think it'll be immunogenic. We'll find out. I think the tox we'll see will probably be exaggerated pharmacology that you will see bleeding. That's where you have to reverse. And then we're moving into larger animals like in pigs on ECMO, et cetera. And then we're also very interested in generalizing or seeing how generalizable this concept of targeting both the exocyte with an Aptamer and an active site of other enzymes is to say can you make very potent now or enhance the potency of a number of small molecule drugs that are out there. And then just acknowledge the people again, highlight shown here in the back is Hai Shan, who did the majority of this work, and then over here on the left side, I want to particularly acknowledge George Pittock, who's a veterinarian in the lab who does a lot of our animal work. So thank you.